How do persister cells make biofilms resistant? Imagine tiny bacterial cells playing hide and seek inside a slimy fortress. These little cells are called persister cells, and they are masters of survival. They hide in biofilms, which are communities of bacteria wrapped in a protective matrix. This matrix acts like a shield, blocking antibiotics from reaching the bacteria inside. But persister cells do something even more clever. They go into a dormant, almost zombie-like state where they stop growing and dividing. When bacteria are active, antibiotics can target their cellular processes, but these dormant cells are practically invisible to drugs. They are like biological sleepwalkers, immune to many treatments. Biofilms form when bacteria stick to surfaces and produce a sticky substance called extracellular polymeric substance. This substance creates a dense, slimy environment that traps nutrients and slows down the flow of antibiotics. Inside this environment, bacteria sense stress and respond by forming persister cells. These cells turn off their normal functions, including protein production and metabolism, making them resistant to drugs that attack these processes. Because they are not actively growing, antibiotics that target cell division or metabolic activity have no effect on them. Genetic systems within bacteria also help produce persister cells. Toxin-antitoxin systems are like molecular switches that turn off vital functions in the bacteria. When these switches are flipped, the bacteria enter a state of dormancy. In Escherichia coli, a common bacteria, certain toxin genes increase the number of persister cells. This genetic regulation is like a survival manual that bacteria use to outsmart antibiotics. Persister cells are not just hiding, they are also reservoirs of bacterial populations. They can wake up when antibiotic levels drop and repopulate the biofilm. This ability makes infections stubborn and hard to treat. Scientists are exploring ways to target these cells by using drugs that activate bacterial enzymes called proteases, which can kill dormant cells. Others are working on enzymes that break down the biofilm matrix, exposing the bacteria to antibiotics. In the world of bizarre biology, persister cells are like tiny zombies that outsmart medicine by playing dead and hiding in slimy fortresses. Their ability to survive antibiotic attacks makes biofilms some of the toughest microbial communities to eliminate. Understanding how these cells work helps scientists develop new strategies to fight persistent infections and clear these microbial bunkers once and for all.